Standard healthcare operates on a baseline of clinical averages. For decades, doctors have relied on population data, treating diseases based on what works for the majority of people. We group millions of people together to find common denominators and establish universal treatment protocols. The trade-off is simple. If your specific biology steps outside that average, the system stumbles. You face delayed diagnoses, medications that simply do not work for your body, or entirely missed windows to prevent a disease. This comparison matrix outlines the shift away from that system. Whole genome sequencing replaces the broad population average with your highly specific biological blueprint. Medicine moves from reacting to symptoms to predicting outcomes based on your DNA. But knowing the science is shifting doesn't make it easy to navigate. The market is saturated with complex terminology like pharmacogenomics and multi-omics, leaving you to figure out which tests are actually worth your investment. To avoid wasting money, we need to map these genomic tools to 3 specific phases of your health: prevention, prescribing, and oncology. Extracting real value from precision medicine requires looking past consumer novelty kits and matching your exact health reality to the correct clinical-grade tool. Let's start with the proactive phase. For healthy individuals, the goal is to map genetic predispositions long before any physical markers or symptoms emerge. The input here is clinical-grade whole genome sequencing. Doctors use this data to spot single nucleotide polymorphisms and high-penetrance mutations, like BRCA 1 and 2 in oncology. You do have to weigh a heavy trade-off, confronting potentially stressful genetic realities up front. In exchange, you gain the ability to launch aggressive, highly targeted screenings. This checklist outlines the strict path to initiate this testing safely and avoid low-quality, direct-to-consumer DNA kits. First, document your family history. Second, consult a genetic counselor. Third, request clinical-grade whole genome sequencing. That middle step is your critical bottleneck. A genetic counselor ensures the massive amount of data pulled from sequencing is interpreted correctly and tied to your clinical reality. For the proactive optimizer, the financial and emotional costs of sequencing pay off. It moves your timeline from managing a disease after it arrives to preventing it from taking hold at all. If you are actively managing a chronic condition like depression or cardiovascular disease, you likely know the exhaustion of cycling through multiple medications to see what works. Pharmacogenomics maps specific enzyme pathways, primarily the CYP450 family, to predict exactly how your body will process a drug biochemically. This flowchart tracks how a standard dosage yields 3 different outcomes depending on your enzymatic speed. If you're a poor metabolizer, the drug builds to toxic levels, triggering severe reactions. Conversely, an ultra-rapid metabolizer clears the drug too quickly, resulting in 0 therapeutic effect. Demanding pharmacogenomic testing requires a slight upfront delay and an added cost before you get your prescription, but it entirely bypasses months of psychological trial and error for SSRIs and avoids the internal bleeding risks associated with blood thinners like warfarin. Testing transforms standard prescribing from an educated, localized guess into a biochemically guaranteed match. The highest stakes application of this science is in oncology, where the approach to treating tumors is actively being rewritten. Instead of attacking a tumor based solely on where it physically lives in the body, oncologists now sequence the specific somatic mutations driving its growth. This graphic demonstrates the difference using lung cancer as the baseline. Rather than broadly blasting the organ, doctors identify a specific molecular pathway, like an EGFR mutation, and block it directly. Sequencing somatic mutations narrows your options to highly specific targeted biologics. You have fewer choices, but the treatment accuracy and tumor suppression are drastically higher compared to generalized therapies. Doctors pair this with advanced monitoring tools called liquid biopsies. Tracking circulating tumor DNA in your blood plasma allows them to spot emerging drug resistance early without requiring invasive surgeries. These tools grant physicians real-time adaptability, preventing you from wasting critical time on therapies a tumor has already outsmarted. Every technology discussed so far relies on your foundational DNA code. But that genetic baseline is static. It cannot show what is actually happening inside your body right now. To understand the true scale of human metabolism, we have to look at the immense biological activity occurring on top of that genetic baseline. This 3D model illustrates the framework of multi-omics. It layers dynamic, real-time shifts, like active gene expression, circulating proteins, and gut microbes, over your static DNA. By processing these massive datasets, artificial intelligence generates a digital twin, a real-time biological replica of the patient. The reality check is that this requires highly complex datasets and massive AI processing power. This is not a service available at a local clinic today. While multi-omics is currently inaccessible for routine consumer care, it points to the vital future of medicine: computational modeling capable of predicting highly effective interventions before a single physical drug is administered. Your DNA acts as a highly individualized biological blueprint, but a blueprint is useless without a builder applying it to a specific problem. This quadrant chart maps these technologies by their barrier to entry and their clinical payoff, creating a clear action plan based on your current situation. If you are healthy but have a concerning family history, invest in targeted whole genome sequencing screening. The emotional input is absolutely worth the preventative output. If you are starting SSRIs or anticoagulants, demand pharmacogenomic testing. Refuse to accept standard trial-and-error prescribing methods. If you are facing a cancer diagnosis, strongly advocate for somatic mutation sequencing and liquid biopsies to ensure your treatment is targeted and adaptable. Precision medicine has moved past the realm of luxury biohacking. Utilizing the correct tools for your specific health stage is now the definitive standard for avoiding the dangerous blind spots of the clinical average.